All right, I'm going to be wild and crazy today and switch to this side just because that's the way it was set up when I came in. So I don't think I'll shock anybody. So remember, exam on Thursday. A few people asked me about that. I'm not sure what the confusion was, but exam 3 on Thursday, that's going to cover chapter 9, which is the antimicrobial methods, chapter 10, antibiotics, and what we're going to finish up today, chapter 14. So it's really just some kind of terms that we're going to go through today to finish up chapter 14. So I'm not going to spill in today too much. Homework due on Thursday as well at exam time. So just 3 chapters, 9, 10, and 14. All right, so see me if you have any questions. I will be in my office for a little while after lecture. If you want to see me, I have a lot of office hours tomorrow, so don't be afraid to come see me and we can talk about anything you want to talk about. Okay, so where we left off last time, we had finished with the portals of exit. And then we were thinking about, well, what happens with those pathogens when they exit a host? Sometimes they go straight to another host. We'll talk about how they're transmitted in a minute. But sometimes they're going to kind of be maintained in the environment somewhere in places that we call reservoirs. Okay, and a reservoir can be an animal, it could be another human, and it could be a non-living reservoir. So when we left off last time, we were talking about animal reservoirs. There's many examples of that. We talked about bats briefly. They are reservoirs for many different infections, mostly viruses. Anytime a pathogen is maintained in some animal reservoir, we call that infection a zoonosis. Okay, so a zoonotic infection is one where, again, it's maintained in animals. Oftentimes the animal is not harmed by the pathogen, but that's not strictly the case. And then as humans, we contract that pathogen by some kind of close contact with the animal. Sometimes we're bit, sometimes just handling animal feces. There's been many cases of people getting rabies just from mucking around in bat caves. It can be through some insect sometimes. So for example, Lyme disease is kind of passed back and forth between animal reservoirs like the white-footed mouse and a tick. Either a white-footed mouse or a deer are typical reservoirs, but then the tick kind of passes it back and forth. And we as humans can inadvertently kind of step into that transmission process. Okay, so oftentimes zoonotic infections are quite serious when we get them as humans. A lot of the emerging infections that we've seen in the past 20 years or so have been what we term spillover events. So we get a pathogen that normally was just passed around maybe some animal host, kind of changed just a little bit, and now it's able to infect us as well. So that certainly was the case for SARS coronavirus, and perhaps the case for SARS-CoV-2, COVID-19 as well. We're not really sure about that yet, but that's potentially. Reservoir humans, some infections as humans can act as a reservoir. So, you know, in the strict sense, that means that you or I, we're not sick with that pathogen, but we can still pass it on to somebody else. There's a case in your book here, a little case study there on page 419 that talks about typhoid. So that's an infection that's typically maintained in water, but sometimes if a person is sick with typhoid fever and they recover, their gallbladder is still infected and can shed the bacterium even though they're not showing symptoms anymore. So this little case study talked about a man in the 1930s that was working to help lay some new water pipes for a town. Apparently he habitually urinated at his job site, perhaps right into those water pipes that fed the water supply to the town. over 40 people died in the town of typhoid until they traced it back to that man who was a carrier for typhoid fever, meaning his gallbladder was infected. Okay, and of course, Typhoid Mary, she's a very famous carrier that wasn't sick herself, but was getting other people sick because she was still shedding the virus. Okay, so there are some cases where humans, whooping cough, we can carry whooping cough, and then when we're in contact, with a baby that's more susceptible to it, we can pass it on to babies. So it's important for caregivers of babies to be up to date with their whooping cough vaccination. Reservoirs can also be non-living as well. So some examples, soil, some things like anthrax spores can be maintained in the soil and just kind of stay there indefinitely till humans or livestock ingest it somehow. Food is sometimes considered a reservoir. Foodborne illness, certainly we can get sick from pathogens in food. As well as water. Something like Cryptosporidium, that's a protozoa that's maintained in the water, and then we ingest it and we get diarrheal disease. Okay, and then if we think about how is that pathogen transmitted to us from some reservoir? Sometimes a pathogen can be transmitted by a number of means, but again, we like to kind of throw labels on the way things are transmitted. It can be considered contact transmission. So the first thing you think of, some kind of direct contact, with a reservoir. It can be just as simple as shaking hands with somebody that has a cold, okay? And then, you itch your eye or whatever, and now suddenly that pathogen, the viral pathogen, has access to a nice portal of entry there. Indirect contact transmission, typically that's going to be by some fomite. And a fomite is a non-living or inanimate object. Okay, so we call that indirect contact because you don't even have to be anywhere near, say, the person or the animal or whatever that was acting as a reservoir. An example of a fomite might be a computer keyboard in a computer lab. Okay, maybe somebody was in there a couple hours before, sneezed on the laptop, had a little bit of flu or something. You came in a couple hours later, touched the keyboard, again, itched your nose, rubbed your eye, whatever. And now suddenly you are sick with that pathogen indirectly through some non-living object. Lots of things could be fomites, it could be, a dirty tissue, or you can probably think of a lot of different things. A water bottle, certainly a glass. Droplet transmission, it's another form of contact transmission when we're exposed through sneezing or coughing. Okay, and usually, that can be just, speaking, laughing, even just breathing. Sometimes those small droplets of moisture that we exhale, that's a perfect way for bacteria and viruses to hitch a ride. The old unstifled sneeze over here, that's a nice illustration. Sometimes those droplets are really small and they stay suspended a long time. So droplet transmission is considered sneezing or coughing closer than 1 meter. Okay, so 1 meter is about 3 feet away. And we think the droplet transmission, that's the most common way that COVID is transmitted. All right, so that's why we all have masks on right now. Why have they always said stay 6 feet apart? Well, because to begin with, we didn't really know. And then we just have a little, we err on the side of caution, I guess. So we think, though, that you usually have to be pretty close to somebody to transmit COVID. Sometimes, though, COVID is considered airborne. So this is considered vehicle transmission. Okay, so air can be a vehicle. So that's when you have really tiny, tiny droplets that stay suspended for a long period of time. And we know, and so that's considered, if you're more than 1 meter away from somebody and you can still get sick, so that's considered airborne. So this happens with COVID occasionally, but we think not as often. Okay. Other infections that are airborne, tuberculosis, influenza to some extent, usually you have to be pretty close to somebody to get flu as well. Waterborne. So sometimes water and food are considered reservoirs. Sometimes we consider water and flu a vehicle. It really just depends on the amount of time that the pathogen spends in that medium. So if the pathogen, lives for a long time in the water, we would consider a reservoir. If maybe, only for a short while till, it's really more of a mode of transmission, then we would consider it that. So again, depends on the amount of time, specific pathogen, as to whether we could consider those things a vehicle or a true reservoir. All right, the third mode of transmission is via some kind of vector. Okay, and a vector is going to be a living organism, almost always an insect. I couldn't find any examples of something considered a vector that wasn't some kind of insect. Okay, so they're living organisms, usually insects, that carry pathogens. Okay, so there's two ways that we could describe those vectors. A biological vector, that just means that the microbe is growing and developing inside the insect. So you would have to know something about kind of the life cycle of the disease that you're talking about to understand if that insect was a biological vector. So there's part of the life cycle of the pathogen in the insect, or whether the vector is just a mechanical vector, meaning that it's just a passive carrier. Okay, so the best example I can think of a mechanical vector is maybe a housefly that lands on the manure pile in the farmyard and then flies to the picnic on the potato salad and maybe carries some intestinal pathogen from the manure now onto the potato salad, and it grows and divides there. So it's just that the pathogen is just kind of hitching a ride. Many examples of biological vectors. Mosquitoes. We talked about malaria a little bit, the life cycle of malaria, how part of the pathogen has to grow and divide within the mosquito in order for it to be, transmit the pathogen back to us. Mosquitoes are pathogens for lots of things. Sandflies, are vectors. Sandflies are vectors for leishmaniasis. It's a protozoa that causes leishmaniasis. Tsetse flies, vectors for several different things, sleeping sickness. Incidentally, it was on the news a few weeks ago that the first malaria vaccine has been approved for use, which was kind of surprising to me because that Plasmodium, there's like 1,000 different varieties of Plasmodium that cause malaria, different strains of it. So I had read that we had thought that was going to be impossible to ever have a malaria vaccine. It's not a great one. It has to be given in 4 doses to young children, and then it only has like a 30% effectiveness rate. So it's not a great one, but apparently there's more in the pipeline. So that's kind of an exciting thing to really kind of dampen down that burden of something that, kids especially can get over and over again. So that's kind of exciting. Okay, but lots of diseases, again, depend on some kind of vector. Oftentimes that's a mosquito. All right, and I just wanted to draw your attention to this table in your book, not to memorize any of these infections, just to get you kind of used to the terminology. All right, so it talks about different diseases, the causative agent, whether it's a virus or protozoa or whatever, the animal reservoir, and then the mode of transmission. And again, sometimes the transmission is complicated. Sometimes it's this way and sometimes it's this way. But I think it kind of gets you a little bit familiar with some of the terminology there. And again, practice using those words. Just to wrap up here, I want to talk about a little terminology that we use, especially when we're thinking about epidemiology. All right, so hot topic these days, the study of where and when diseases occur. So it's good to be fluent in some of these terms. So you've heard many times before. But the difference between incidence and prevalence, okay, they kind of sounds like the same thing. When we talk about incidence of disease, that means the number of new cases in a given time. And then the prevalence is how many people in a given time are living with that disease. Okay, so the incidence is never going to be higher than the prevalence. Okay, incidence, new cases, prevalence, number of total people. So let me skip to this graph that they show you in your book. And I left off the title of this graph. This is the incidence of AIDS in the US, incidence and prevalence. And this graph is a little bit confusing because the units on the incidence side are different than the units over here on the prevalence side. So when you look at this graph and you see, oh, the incidence of AIDS, the number of new cases peaked in the US somewhere around 1993, 1994. It peaked at about 80,000 cases in 1 year. The prevalence at that time, again, you got to look at these units over here, was somewhere around 120,000. Okay, so again, it's confusing with the units there. Interestingly, so the incidence for the most part went down as we had better, education about what was causing it. Meanwhile, the prevalence is skyrocketed. What can explain that? Well, people are living longer with AIDS now. They have almost a normal life expectancy as long as they stay on their AIDS drugs. So while the incidence is low, we are still getting new cases of AIDS. People aren't dying from it, so we have a higher prevalence. And then a few other terms that we use to describe occurrence of disease. Diseases sometimes are called sporadic. And that just means you have a few cases here or there. That's not very common, and the cases don't seem to be connected with each other. I was trying to think of an example of a sporadic infection, maybe something like botulism. Okay, so botulism is a foodborne illness. People don't typically pass it around. I mean, they can't pass it from one person to the next. It doesn't happen very often, and a case shows up here or there. An infection that's endemic means that it's constantly present. So every year, we have flu, we have colds, we have strep throat. We're constantly going to have them. They may fluctuate with the season up and down a little bit, but they're always with us. An infection that's epidemic, we're getting an unusually large number of cases. And then a pandemic is an epidemic in one or more continents. Okay, so how do you know? The question is, how do you know if you have an unusually large number of cases? Well, the CDC keeps track of this sort of thing. So I included a link here to the charts that the CDC keeps track of every week. And there's a picture of this in your book as well, but I just took a screenshot of part of it here. So these were the nationally notifiable infectious diseases for week 40. That was just a couple weeks ago when I was preparing for this. And it used to be the CDC would actually publish a mortality and morbidity weekly report, the MMWR. They published this newspaper and they would send it out to public health officials, every week. Now they just post it. But so there's all these lists of diseases that if you go to the doctor and you're diagnosed with, that your doctor has to notify the CDC that you have that. So something like, I just picked one out that maybe you've heard of before, botulism, for instance, and I clicked on that, and here's the table for botulism. Okay, so this is one that's not very common, but you can look it up by state, how many cases are in the states, or let's look at all of the US. So for this week, there was no cases of botulism. In the previous 52 weeks, what was the week that had the most number of cases? There was 2 in one of the previous weeks. And currently, so far, we've only had 11 cases of botulism in the US. Compare that to last year, at this point, there was 8. So pretty close, nothing really to worry about there. And this week and in the entire US, there were 4 cases. So, whoops, we started back over again. So there's different kinds of botulism. There's foodborne, infants, and others. So anyway, so that's how we kind of can keep track of, if suddenly we had in 1 week, 400 cases of botulism, oh, okay, we need to send some people out there and figure out what's going on. All right, so that's how we kind of know whether to consider something endemic or epidemic or sporadic. Okay, another picture from your book that kind of helps illustrate those things, endemic, always with us, sporadic, a few cases here or there, epidemic, an unusually high number, pandemic, epidemics on more than one continent. And just to point out that, something that's considered endemic doesn't necessarily mean there's only a few of them. So in this case, we had an endemic infection here. That's the blue line up here at the top. So it kind of fluctuates with the season, and the actual numbers here are in the purple. Kind of fluctuates up and down. But we always have something like flu with us, where a disease can be considered epidemic. There's way fewer than that endemic disease, but if you have, a much higher than the normal amount at any one time, then it's considered epidemic. Okay. All right. So that is all I have to say about chapter 14, an infection. We didn't go too far into today, so that's good. Any questions there before we move on? Yeah. Yes, thank you. So that's the last material that's covered on Thursday's exam. Okay, from now, the immunology stuff, that will be on exam 4, which is right before Thanksgiving break. So not too far away, but. All right, so chapter 15 and 16, immunology, this is kind of getting into material that's usually a little bit more conceptually difficult. A lot of you have had some exposure to innate immunology. That's what chapter 15 is. Chapter 16 is getting into adaptive immunology, which is usually a little bit newer territory. So just as by way of an introduction to both chapter 15 and 16, why do we need an immune system? Well, it protects us, according to the CDC, from a wide range of pathogens and parasites and, parasitic worms. So thank goodness for our immune system. It's also necessary in order to repair any wounds that we get, to eat up dead cells that we don't need anymore. But sometimes it gets a little bit out of whack and it has the potential to cause issues itself. Okay, so that's what autoimmune disease is. And it gets complicated. Okay, I didn't post this. I don't want to scare anybody, but it's our immune response. It's multifaceted and it overlaps quite a bit with itself. So that makes it more difficult to learn about. Anytime, okay, we want to learn what macrophages do. We can't really learn about macrophages without understanding the signaling that goes on from other kinds of cells and without thinking about what B cells do and T cells do. So again, it kind of makes it hard to know where to jump in and start. Sometimes you'll hear me talk about something and then say, don't worry, we're going to talk more about that later, and I'll explain that later. But you know, and it's a pretty new science, so we're still learning a lot about our immune responses, so that makes it a little bit more complicated as well. So basically everything that we talk about, it's actually more complicated than what we're talking about. All right, so keep that in mind too. But the way we're going to come at it, okay, we're going to try to classify our immune response into 3 different lines of defense. And these are totally artificial constructs. So I'm not going to say, oh, is our macrophages, are they, which line of defense are they? They're fairly artificial, but usually that first line of defense, once those things are breached, then the pathogen is going to encounter the second line of defense, for example. So we'll start with the first line of defense. Just an overview here for a minute. External physical barriers, and this is part of our what we consider our innate immunity, and that another word for that is non-specific. Okay, so basically the next time that same pathogen is encountered, our body's going to respond the same way when things that are considered innate immunity. So things like skin and other bodily processes that we don't necessarily consider part of our immune system actually have some immune function. So we're going to talk about that a little bit. Some of our defenses and mucosal membranes and the role that our normal microbiota may play. We're just starting to learn about that. And just kind of briefly mention some other processes there. The second line is considered cells and processes. This is also considered innate immunity. So again, our responses are fairly non-specific. The traditional way to think of that is, again, the same response the next time the pathogen is encountered. I have been reading a little bit about how there is some slight tweaking that does take place, but again, traditionally, pretty much the same response. So we'll look at the role of white blood cells and things like complement proteins and inflammation, fever, the role that those things play. And then the third line of defense, this is considered part of our adaptive immunity. So incredibly kind of tailor-made specific response to pathogens that are encountered. And that's either considered cell-mediated or humoral, and that means in our bloodstream. Okay, so the action of antibodies and cytotoxic T cells that are going to kill parts of cells that are infected. All right, so that's just a little overview. So a fun question to think about before we get started here. I don't know if you've ever played the game Would You Rather. Okay, it's a game where they say, oh, would you rather eat a cockroach or a salamander, or something like that. All right, so if you had to do without one branch of your immune system, which would you choose? Your innate immunity or adaptive immunity? So just with that brief overview that I gave you, there actually turns out there is a correct answer here, a way that we think that you'll live longer. So just turn for a second to your neighbor and say, I would rather do without my innate immunity, or I would rather do without my adaptive immunity. Okay, so did you agree with your neighbor, or did you have different ideas? When I've asked this as a clicker question in the past, it was usually pretty 50-50 which one the students would pick. So it turns out you would live longer if you were missing your adaptive immunity rather than your innate immunity. Okay, so here's the experiment that shows evidence of that. So it's kind of interesting. We can do amazing things with mice. We can knock out a mouse's ability to do like certain processes or to make certain cells. We call those knockout mice. So we can really just do some really cool things. So they exposed mice to some infection, and I don't know what they used in this experiment. So normally, so here's they exposed it to the infection, some pathogen. Normally, the numbers of the pathogen are going to rise up, and then, you know, eventually the immune system takes hold of it and clears the infection. Okay, but here they took mice that were SCID mice. That means severe combined immunodeficiency mice. These were mice that were not able to make B and T cells, T and B cell negative. They could not make those. They could make PMNs, that's another name for neutrophils, and they could make macrophages, two cells that are really important in the innate immunity, but not T and B cells. So you can see with the innate immunity, they could kind of fight off the infection a little bit, but not really totally clear it. Okay, so ideally you're going to have both adaptive and innate immune cells. But here, up here, the red line, these are mouse that are lacking innate immunity. So they don't have neutrophils, are not able to make macrophages. Basically, unbridled reproduction of that pathogen. It turns out that you need those innate cells in order to kick in your adaptive immunity. So even though they can make B cells and T cells, those don't get the right stimulation to act. And that mouse is not going to live very long right there. So it turns out that we are going to live a little bit longer if we're, you know, lacking adaptive immunity, but obviously we want to have both. All right, so it's kind of not a good situation either way. All right, so let's start looking at some of our first lines of defenses. We talked about skin as a possible portal of entry for pathogens, but we said it's not really a very good portal of entry because our skin is a really effective barrier. So let's look at some of the things that make skin such a really effective layer. You have several layers of skin. The outer layer of skin, what we would call our epidermis, it's made of many layers actually, but the outer layer, we've got many cell layers, and the outside layers are dead. Okay, so if you look at this scanning electron micrograph of our outer layer of skin, okay, those look like dead cells. They're dry, they're flaky, and they're continually being sloughed off. Okay, so continual, we call that desquamation, meaning we are continually losing layers of dead cells. Okay, so you might wonder, well, where do all these dead cells go? Well, they're part of what makes up dust in our house. And luckily, there's things that feed on our dead cells. They're called dust mites. And unfortunately, about a third of us are allergic to not dust mites themselves, but the feces of dust mites. All right, so if you have an indoor allergy, then you're probably allergic to that. But you know, thank goodness we have something that's eating up our dead skin, because it would be piling up around us if we didn't. Okay, so many layers, they're tightly packed cells, and we're continually losing the top layer, so that pathogens, when they have to adhere to their target cells, they're just continually being lost there. Deeper in the skin, there's lots of defenses here. They contain a particular kind of phagocytic cell that we're going to talk more about, dendritic cells, patrolling around for pathogens. We have sweat glands that secrete sweat. And sweats can be fairly salty. Okay, so we know that organisms typically don't like to be in the presence of high salt because it makes their cells lose water, bacteria anyway. Sweat also contains antimicrobial peptides. And I'm not going to get too specific about what those are. We're just going to kind of gloss over that. But there are certain things in sweat that are going to reduce the chances that bacteria are going to grow, as well as lysozyme. Remember, lysozyme is a enzyme that's going to disrupt the structure of peptidoglycan. So we're going to see that quite a bit in some of our bodily fluids. Okay, so it's going to act on bacterial cell walls. All right, so here's a sweat gland here. We also have sebaceous glands in our skin, and they secrete sebum. Okay, so that's what this arrow is right here. This is considered a sebaceous gland. Sebum is what helps make our skin pliable. Again, that keeps it an effective barrier. It also is going to lower the pH of skin. Okay, so low pH, salty environments, some other antimicrobial chemicals and cells in there makes it a pretty inhospitable place. Okay, sometimes we get some hair follicle infections here, but for the most part, as long as the skin is intact, it's a pretty good, not a very good portal of entry. You'll remember that mucous membranes are a much more common portal of entry, especially we said, remember respiratory tract is the most common portal of entry. We do have defenses there, but they're just a little bit more easily breached by pathogens. Okay, so mucous membranes, well, I think I spelled that wrong. I think there's no U in that actually. You would think there would be, but I think that's not right. Line the respiratory, urinary, digestive, reproductive tracts. There are layers of epithelial cells. Epithelial cells, I remember, they're the kinds of cells that line sort of the outside of our body, where our body meets the outside. That layer is thin. It's tightly packed together, and the cells are alive. We still have desquamation taking place even though those cells are alive. So again, the desquamation, we're continually replacing those cells. So pathogens do attach, they're basically sloughed off. There are stem cells deeper within the epithelial that are going to give rise to a couple different kinds of cells lining. We'll talk specifically about our respiratory tract here. There are goblet cells. And your book doesn't give a picture of this. This was the best graphic that I could find here. But goblet cells, these are the ones that secrete mucus. And mucus is going to be important to trap pathogens and then eventually move those trapped pathogens along kind of out of the way from their target cells. And what I've read that we produce about 1 liter of mucus a day. So that seems like an enormous amount. So not just snot is mucus, but again, and not saliva. That's something different than mucus. Our body just creates a lot of that. It's really important to keep our mucous membranes moist, and again, to try to help keep pathogens kind of from attaching. There are also the second kind of cells that we see here lining the mucous membranes are ciliated columnar cells. Okay, so again, this is on the respiratory tract only. We don't have cilia lining our other mucous membranes, but they are going to, here's a columnar ciliated cell. Going to give rise to cilia on its surface. So along the respiratory tract, again, we have that cilia that moves in kind of a wave-like manner that helps move the mucus and the trapped pathogens out of the way. Incidentally, if you are a smoker, Oftentimes people that smoke, their cilia doesn't move very well. And eventually people that smoke, there's changes made in these cells so that they can no longer produce cilia. So smokers are typically more susceptible to respiratory infections because they're not as good at, again, keeping pathogens from attaching. And there's a lot of evidence, although I haven't seen any really big studies come out about this, that smokers are more vulnerable to COVID as well. Smoking does kind of dampen certain kinds of immune responses as well. So also within those mucous membranes, there's some antimicrobial peptides. And again, I'm not going to spend too much time talking about those in particular, but things like lysozyme are present. There's also going to be dendritic cells patrolling. Again, this is a kind of phagocytic cell that's going to go around and try to engulf pathogens. All right, so we do have some defenses there in our mucous membranes. Also want to consider our normal microbiota as actually part of our immune response, which seems a little bit crazy, but we've mentioned this before, how there can be some microbial antagonism. Meaning if you already have bacteria in lots of places of your body that we've kind of come to terms with, or our immune system is kind of allowed to be there somehow, any new pathogen, it's going to have to compete with the microbes that are already there for space and nutrients and oxygen or whatever else. Okay, so our microbiome outcompetes more pathogenic species. Okay, so we've talked about that in terms of, or maybe I talked about it in lab, I forget. I think I gave that example in here, how Staph epidermidis that lives on our skin probably outcompetes other organisms that would try to gain a footing there. We also know that our normal microbiota throughout our body plays some role in stimulating our immune cells. And we know this because, again, they've done some really cool experiments with mice. They've raised mice in sterile environments, which, man, that would be a lot of work to do that, but they birth them from mothers and anyway, just keep them in cages where everything's been sterilized, their food is sterilized. So essentially they raise mice that have no normal microbiome. And what they've observed about these mice is that they don't grow very big and they don't grow very fast. And if they are introduced to some bacteria, they often don't have much of any immune response to them. So organisms that wouldn't normally kill a mouse are often cause deadly infections in mice that don't have normal microbiomes. So we know that our microbiome plays some role in keeping our immune system, our immune cells primed and ready for action. And we know that our microbiome makes other contributions. We still have a lot to learn about this, but some examples, we know there are bacteria that produce vitamin B5 for us. We get a little extra folic acid from our bacteria in our gut. We kind of get their extra, what they make. We get most of our folic acid from our diet. So that's why when we take a sulfa drug that would inhibit that metabolic pathway, that doesn't affect us because that's affecting that metabolic pathway in bacteria. And bacteria also provide for us a precursor of vitamin K, which is necessary for us to metabolize glucose. So we know that we do get help from our normal microbiota, and I think that's an exciting area that we'll have a lot more to learn about as well. And lastly, with the first line of defense, there's just some nice tables in your book here, not to memorize, but just kind of make you aware that a lot of our body processes that we don't normally think about as being part of our immune system definitely has a role in helping out, help defend our body. So if you look at some of the secretions, saliva, for instance, has a lot of antimicrobial chemicals lysozyme, we keep mentioning that as part of saliva. And of course, we think about saliva as being necessary to digest our food, to help, wash it down into our stomach. But there are lots of antibacterial enzymes in there. And here's a practical point. When we go to sleep, we produce much less saliva in our mouth, which is a good thing. Otherwise, we drool all over our pillows, right? But it's really important to brush your teeth before you go to bed. So if you only brush your teeth once a day, do it before you go to bed, because there's so many antimicrobial chemicals in your saliva. And when you're not producing those as much at night, our bacteria are going to overgrow at night. So you want to, get rid of as much bacteria before you go to bed, so you're not having as much trouble tooth decay, hopefully, in the long run. Stomach acid, of course, is going to inhibit microbes at low pH. Most of them can't survive that. So a lot of, just actions that are, we're moving fluids out of our body. They're going to be important to contribute to first line of defense. So that lysosome, we see that in several bodily fluids. The other kind of hallmark of thing that we see is lots of our secretions have mechanisms enable it to sequester iron, meaning we're trying to, iron is often a limiting nutrient for the growth of bacteria. So a lot of our, we have chemicals to try to sequester any available iron so that bacteria can't have access to it. So we see that in several types of secretions here, binding iron, just to try to limit that. Okay, so again, those kind of first rounds of defenses are just kind of mechanisms that are already there in our body, kind of physical and some chemical mechanisms. When that first line is breached, then typically we start thinking about our second line of defense. So this is also considered innate immunity. And I don't know if I posted this, but it's just kind of a rehash of what I posted before. All right, so we're going to go through all these things individually. We're going to spend a good bit of time thinking about our immune cells. And this is probably something that's a little bit familiar from anatomy, maybe if you've had it, till we get into some of these other processes like complement and inflammation. All right, so this is, let's see, did I not? There it is, figure 15.4 from your book. Okay, talking about the cells that are present in our blood and thinking about, well, kind of where they came from or how they originated and what they do. We're going to talk about them all specifically. But we all have stem cells in our bone marrow that give rise to different kinds of precursor cells. 3 different kinds: erythroid, myeloid, and lymphoid precursor cells in the bone marrow. And that process of differentiation is called hematopoiesis. Okay, so again, it's just a process of our stem cells being differentiated into more specific kinds of cells. Okay, so those erythroid precursor cells are going to give rise to red blood cells. Okay, of course, what red blood cells do, they carry oxygen. And they don't really have any immune function that I'm aware of, so we won't really mention those anymore, but obviously they're really important to get oxygen to our tissues. And then myeloid precursor cells are going to differentiate into lots of different kinds of cells. Platelets, again, present in your blood. The main function of platelets is to help clot the blood. There is evidence that platelets can have a little bit of an immune function, that they may, when pathogens bind to them, they can release some antimicrobial chemicals, but their major job is clotting blood, so we're not really going to talk about those very much anymore either. Okay, now we get into our white blood cells. Here are basophils, neutrophils, all the phils, and eosinophils. And you don't need to learn how to recognize those. I don't know how to do that either. You'd have to take a histology course to really get good at that. They are named based on what kind of dyes they take up, I think. But these three kinds of cells, these are considered granulocytes because they have these granules filled with various inflammatory mediators and digestive enzymes. Okay. And then the other kind of cell that originated from myeloid stem cells, these are monocytes. And then when they leave the blood and migrate out into the tissues, they give rise to two different kinds of cells, macrophages and dendritic cells. Okay, so we'll talk more about those shortly. And then lymphoid precursor cells are going to develop into lymphocytes. Okay, and some examples of lymphocytes, B cells, T cells, and natural killer cells. Okay, we usually abbreviate those and call them NK cells. Okay, we're not going to spend much time at all on natural killer cells. We'll spend a lot of time on B cells and T cells. But these monocytes and lymphocytes, they make up the agranulocytes. A meaning without granules. Okay, so you got your granulocytes and your agranulocytes. All of the granulocytes and agranulocytes, those are also called leukocytes or white blood cells. Okay, we've got a couple different names for those, leukocytes or white blood cells. So it's a lot to keep track of, all the different cells that are made there, but we're going to look at them one by one so that we can kind of keep track of them a little bit better. And before we do that, when I was going through my lecture notes yesterday and thinking about hematopoiesis, I got an email advertising this week's seminar. So we haven't had, I think we've had one other seminar this year and no other ones in the last couple of years. So it's kind of exciting. We're getting back to our seminar series. But the title of this week's seminar this Friday is right here: specific W and T signals are required for hematopoietic stem cell development. I was like, oh, there's that word, hematopoiesis again. So our talk on Friday is going to be how things can go wrong with that differentiation of stem cells into other kinds of cells and how that can cause cancer. Okay, all that lots of signaling that happens in that hematopoiesis process. So if you're interested, or you think maybe someday you would want to go to graduate school and do research, you should go to some of these seminars because it helps you kind of understand how that research process works. Even though they very quickly get off into things that, you know, even though I'm totally lost and have a little bit more background. But again, just a little plug for our seminar that's noon on Friday over in Manitou 107. So just thought I'd stop and give a little plug for that. Okay, so let's think about what some of these immune cells do. And again, I put a little picture over here of them. Don't worry about identifying them. But here's a neutrophil here. You can see the white blood cells are generally a little bit bigger than the red blood cells. And I wanted to talk about neutrophils first because they are the most common kind of white blood cell in circulation. So 50 to 60% of white blood cells in the bloodstream, in other words, in circulation, are neutrophils. So of all those different cells that we talked about, half of them that we find in our bloodstream are neutrophils. Okay, they are also known as polymorphonuclear leukocytes. Okay, I know it's always terribly confusing when you have a couple different names for the same thing. So PMNs. otherwise known as PMNs. Okay, so where did that name come from? Polymorphonuclear. If you look over here at the picture at that purple inside the cell, that's the nucleus. And it's got many different lobes of the nucleus. It's really kind of an odd looking nucleus. Okay, now, as we'll see, basophils and eosinophils also have that weird polymorphonucleus structure. But so technically, PMNs are technically eosinophils, basophils, and neutrophils. But because neutrophils so outnumber those other cells, we often use that term PMN synonymously with neutrophils. Okay, so usually we use those the same. These cells, what else about them? They're highly motile. Okay, so they are actually considered kind of the first responder to a site of infection. Okay, they're the ones that are going to follow some chemical signals, and they're usually the first ones there to try to deal with the invading pathogen or the whatever site of injury. Now they don't have flagella. They are moving usually like an amoeba would. So amoeboid action, extending pseudopods and moving that way. And they're very short-lived. So in circulation, they might live maybe a few days, up to 5 days, but usually in the tissues, they're going to live less than 24 hours. And that actually is okay. We are just continually replacing our supply of neutrophils. We'll see why it's not such a good thing that when they start acting on pathogens, we don't want them to keep acting over and over and more because they actually will do tissue damage if they do that. So we want them to act a little bit, die, and then we'll replace them if we need more. All right, so how do neutrophils act? They actually have 3 different ways that they can work to invaders. Okay, they very commonly act by phagocytosis. So we are going to look more closely at that process of phagocytosis. We probably won't get to it much today, but you know, they're extending pseudopods, they're engulfing bacteria, or invaders, making enzymes in order to basically eat them and kind of exocytosing whatever is left over. They also will degranulate. So remember, they're granulocytes. They have these granules filled with enzymes and other kinds of inflammatory mediators. The granules are not the same as lysosomes that are necessary for phagocytosis, but they have sort of the same kinds of things in them. And then we know that neutrophils act by one other way that really I think was just described in the last 10 or 15 years. They can act by formation of nets. And nets stand for neutrophil extracellular traps. And so you can see this over here, this poor Klebsiella pneumoniae bacterium has been caught in one of these neutrophil extracellular traps. And you don't see the neutrophil in this picture because basically the neutrophil has kind of blown itself up in order to immobilize this pathogen. Okay, so the neutrophils have ways to kind of degrade their DNA, and basically it's kind of like a, I don't know, like a hand grenade or something. They degrade it and then can kind of blow it out of their cell in order to trap pathogens. And if you've ever done like maybe some kind of mini prep in the lab where you've isolated DNA from bacteria cells, it's kind of sticky, DNA is. So the DNA, again, is sticky, and then there's also other antimicrobial peptides associated with that that is just going to act to trap pathogens and keep them from invading deeper into tissues. So this picture here is of a mouse lung, a bacteria invading a mouse lung. Okay, and this picture, I didn't post this. This is always kind of a weird picture to me that I got this from Wikipedia, actually. This is supposed to be a neutrophil that's enveloping an anthrax bacterium. Kind of reminds me of like a python eating an antelope or something, because you know, you would think here that the neutrophil would be way bigger than the bacteria, and that's what doesn't make sense to me about this picture. But it's kind of cool just thinking about how it kind of envelops and eats, basically. And then I found one more graphic, just kind of looking around the web. Again, these are the 3 ways that neutrophils, we know how neutrophils act on pathogens. I'm seeing this term a little bit, netosis. Basically, the neutrophil extracellular traps, how they are degrading their DNA and trapping pathogens in that kind of stickiness there. Okay, so the other 2 kinds of granulocytes, eosinophils and basophils, I decided to just group them together because they're pretty similar in terms of their function. There are very few of them actually in circulation, of maybe a few more eosinophils than what there are basophils. Again, they have similar function. Mostly they act by secreting their granules. Okay, so by degranulation, yeah. Anyway, you know what I mean. Generally, they're not phagocytic. Apparently, eosinophils can phagocytize, but they don't normally do that. I think there's a homework question about that that's a little bit confusing. So they can phagocytize, but it's not the normal way that they act. They don't do the nets. That's a neutrophil thing. And eosinophils are best known for targeting large eukaryotic pathogens. So things like roundworms or flatworms. Those are helminths. And let me I'll come back to this in a minute. I think that's a really cool picture. I didn't post this because I don't have a good citation of where this came from. I actually found this picture on Pinterest. And I thought, man, I do not know what algorithm they are using, but they have me pinned down. I mean, they've got, you know, crochet hats and air fryer recipes, and then this shows up. Right? Yeah, they know me. But it's kind of a cool picture. This is supposed to be a parasitic fluke. And then all these eosinophils are chemotaxing, getting signals to attack that fluke. And so these are all degranulating. So again, it's not going to make much difference if one eosinophil degranulates, but you get a whole bunch of them, and that fluke is definitely in trouble. So we typically think if we find the percentage of eosinophils in somebody's blood is elevated, then we start to suspect, oh, maybe there's some, you know, parasitic infection, maybe by helminth worms. The other possibility is that there's some allergic reaction going on with the patient. Okay, so we tend to see more eosinophils when there's some kind of inappropriate inflammation going on. In basophils, they're not really famous for anything other than kind of doing the same sorts of things that eosinophils do. And there's just less of them, so they kind of play less of a role in all those same kinds of reactions. All right. And then moving on to the agranulocytes. Okay, so these are monocytes and lymphocytes. So I'll start with the monocytes first. Pretty good percentage of white blood cells are circulating, or some of them. Again, like I said, they differentiate into macrophages and dendritic cells. Macrophages are best known for being phagocytic. Whoops, I didn't show you all the pictures there. There we go. So they are going to not only be kind of garbage collectors. So some macrophages are circulating throughout the body and just eating up dead cells. Sometimes macrophages will stay associated with a certain kind of cell, like we have lots of alveolar macrophages, deep within our lungs, or we have lots of macrophages in our spleen. But there's other macrophages that again patrol around, and something that they're really good at is processing and presenting antigen to lymphocytes. So I'm going to get ahead of myself a little bit, but macrophages, like I said, our innate cells are often important in terms of stimulating our adaptive cells. So if you have a macrophage, for instance, so here's macrophage, that circle with a line through it is a abbreviation for a phage. So here's a macrophage, and it's going to extend pseudopods, tied up my drawings, rudimentary, to engulf some pathogen. So here we have the pathogen inside the macrophage in that endosome vesicle, and the macrophage is able to kill that, and then there are mechanisms that we're going to study a little bit more in depth later for that macrophage to present some of that pathogen on its surface. Okay, so usually amino acid sequences, so parts of proteins, are going to act as antigens, meaning it's going to elicit some kind of immune response. So that macrophage is going to present that antigen as if to say, hey, everybody, this is what I'm dealing with. And it's going to call in help from B cells and T cells that have receptors for that same antigen. Okay, so usually those macrophages are going to be flowing through the lymph, where they're going to come in contact with other cells, and then they can get help to fight that specific invader that was originally phagocytized. So when we say process and present antigen, that's kind of what's going on there. And they talk about that a little bit in this YouTube video. It's kind of a cartoon thing that kind of goes through that real fast, but kind of just to underscore what I'm talking about there. What else about macrophages? They are going to communicate with other cells by cytokine signals. And I'm going to talk a little bit more about cytokines later, just to talk about the different kinds of molecules that are considered cytokines. But for now, these are just kind of messengers, signals that cells can send in between each other. And in contrast to neutrophils, macrophages are typically pretty long-lived in terms of surviving for months in our bodies. So, oh, and I forgot to come back to that point. Like when neutrophils degranulate, those granules are containing things that are going to kind of encourage inflammation in our bodies, which we'll get to more about what that means. But it's not, those granules not only act on pathogens, but they act to cause damage in our body as well. So we don't want cells to keep degranulating over and over again. So like I said, we just want them to act for a little bit, and then if more of a response is necessary, we make more cells. So macrophages are not degranulating, they're not causing harm typically, so we can let them live for a lot longer. Dendritic cells are also derived from monocytes. They're going to do a lot of the same things that macrophages do. They're phagocytic, very plentiful in the skin and mucous membranes. We talked about them already there. And they are also processing and presenting antigen. Just like the macrophages. So here are dendritic cells. They are just crazy looking cells. That name dendrite, that comes from the Latin word for tree. So I'm not sure if it's branches or roots or whatever, but they kind of resemble neurons, basically, the way they look branched like that. But we think that that makes them particularly good at presenting antigens, kind of on those long arms or appendages of their cell. All right, and the other kind of agranulates are the lymphocytes. Kind of interesting. I guess you can't really tell the difference between B cells and T cells when you look at them under the microscope, but they have this huge nucleus that takes up kind of most of the cell there. Again, don't need to identify that, but I just thought it was kind of interesting. Makes up a good proportion of white blood cells, but we typically don't have that many lymphocytes in our bloodstream. They're more often located in the lymph. That makes sense, their name. So both B cells and T cells are lymphocytes. So just a little introduction to them. We're going to talk more about them later. The B cells, their main role is that they are going to differentiate into plasma cells. And then the plasma cells are going to make antibodies. Okay, so sometimes we call the B cell response as being part of our humoral immunity. Humors are just bodily fluids. Okay, so when we're talking about humoral immunity, mostly we're talking about in the bloodstream. So antibodies, again, are going to be made and released into the bloodstream. They can leave the bloodstream and go out where they need to in the tissues at certain times. There's several different kinds of T cells. And we'll, again, just an introduction, we'll talk more about this later, but cytotoxic T cells and helper T cells are going to be really important in terms of what we call cell-mediated immunity. Okay, so it's those cytotoxic T cells that are actually important to kill cells that are infected with viruses. Okay, so there's a lot of talk about, oh, antibody titer, you know, once you've been vaccinated for COVID, but really that's only one piece of the puzzle. We really want a good cell-mediated response because the cytotoxic T cells are the ones that, again, act best to kill virally infected cells. And just to mention briefly here, natural killer cells, these actually function as part of innate immunity, where B cells and T cells are considered adaptive immunity, really very specific receptors for different kinds of invaders. But natural killer cells are actually are going to act against virally infected cells. We originally called them natural killer cells because we didn't really understand the signals that they got to know that a cell was infected with a virus and that it was appropriate to try to kill it. It was just a little bit more complicated, and eventually we figured it out, but that's why they have that kind of a strange name, natural killer. All right. I'm going to spend probably 5 or 10 minutes here talking about phagocytosis. So this seems like a good time to stop. So we'll pick up there next time. Again, let me know if you have any questions about the exam on Thursday, and I'll see you then.